This week, our lecture topic's on cell division, and it's an interesting topic. All cells divide. That's one of the primary kind of functions of life, is to create more life. And if we take a look at this picture, we're seeing the nucleus of the cell, and it's starting off. The DNA is kind of unwound. Proteins are being made from it, and as the cell cycle progresses, the DNA duplicates, and then it condenses down into discrete units, and they line up, and we'll take a look at this process in this lecture, and they get half the DNA, right? It duplicates, so half of it gets pulled one direction, the other half gets pulled the other direction, and we end up with two identical copies of a cell. And that process of splitting up that DNA is called mitosis. And that creates an identical cell, like your skin cells would divide, just like this, and most of the cells in your body. In fact, there are some cells in your body that don't divide like this, but go through a process of creating sperm and egg, and that's called meiosis. So that produces sex cells. We'll also take a look at that cell division in this lecture. So let's go ahead and ask a couple questions. Why do cells divide? Philosophically, it's hard to say. In science, we don't know. We don't know how life got started and why, it started dividing and how the first cells formed. It's really, we just don't know about that. But we do know that, we have trillions of cells in our body. So when the sperm and egg fuse to make us, like, a lot of cell division had to happen, right, to produce us. And why do they have to divide accurately? Well, it turns out if they don't, right, we can get something like cancer, which we'll talk about later. We're going to start off just looking at how a bacteria replicates. This process is called binary fission. It's when one bacteria turns into two, and we could say, for instance, be looking at an E. coli bacterium, and there's not a lot of internal structure. There's just the DNA that really needs to be copied. So enzymes will copy the DNA, and then the cell needs to split into two and put one of the pieces of DNA, right? It has one circular piece, so it's pretty simple. Just put one piece in one side, one piece in the other. The cell has to grow larger, and it turns out E. coli can divide optimally in 20 minutes. If we refrigerate our food, obviously that replication takes a lot longer, because we know, what does lowering temperature do on the speed of enzymes? It greatly lowers how fast they work. So the DNA just can't divide very quickly. So if we left food out, right, it could spoil. What would happen if we left some food out and there's bacteria everywhere? So imagine there's 10 E. coli in some food, and you leave it out for 3 hours. Well, if we know the bacteria will divide every 20 minutes, we'll see exponential growth. So it starts off at 10, in 20 minutes it'll be 20. Every cell divides. And then in 20 more minutes, it'll be 40. And then all 40 will divide. After the first hour, you'll have 80. And then we'll see those divisions again. So 160, 320, 640 after 2 hours. And then if I could do math in my head past that, right, it'd be like 1280, 20, 560, and then 5120. And that would be after 3 hours, right? So you see this exponential growth where it just, the population shoots up. And that's why the longer you leave something out, the more likely it could spoil. Okay, let's take a look at eukaryotic replication. So eukaryotic cells are different. They don't have one circular chromosome. They have many linear chromosomes. And during the cell cycle and cell division, the DNA will wrap up tightly into what's called a chromosome, okay? And the DNA usually is unwound, and it'll wind around what's called a histone protein. And there's several histone proteins. It'll wrap up. Here's some electron microscope pictures, and then an artistic picture of wrapping around those like purple histone proteins here. And then it'll be packaged up really tight into what's called a chromosome. And because the DNA has divided so that we can make two cells, we have one, like chromosome number 1, and then its copy, and they're stuck together. And the goal of the cell cycle is to pull these copies apart into two different directions, and to do it in a controlled manner so that we can make two identical cells if it's a cell in our body dividing. Why does the DNA have to wrap up tightly? We can say that we'll take a look at a picture here that will make that a little more clear of like why the DNA gets wrapped up like this. So to do that, let's take a look at the DNA from a human. And all the DNA that we have, we usually call them our chromosomes, okay? And here's a picture of our DNA. We have 46 chromosomes. Number 1, 2, 3, but we only have 23 distinct chromosomes. And we got 23 from our mom, and we got 23 from dad. So we have 23 pairs. So that turns out that we have 46, but we have 23 pairs, right? And each pair is called homologous chromosomes. So we got number 1 from mom, number 1 from dad, and that pair is called a homolog. And the reason they're a pair is because chromosome 1 from mom, let's say for instance, has some genes on it for, let's say, eye color and hair color, but maybe mom gives you, brown hair and brown eyes, and then dad has the genes for, blonde hair and blue eyes, right? So that would be on these chromosomes. And the genes, for hair color or eye color would be on that same chromosome, but there could be variations. So we get a full complement of DNA from mom, a full complement from dad, and we end up with 23 pairs. And they go from biggest to smallest, and then number 23, the final pair, are sex chromosomes. So XX is female, XY is male. Okay, and to answer our question is why does the DNA wrap up into these discrete chromosomes? In a cell that's not going through division, the DNA is unwound so that protein can be made. And if you tried to pull apart DNA like that, they're so long that they would get intertangled and it'd be a big mess and the cell would just get stuck. So during the cell cycle, the DNA gets wrapped up tightly into chromosomes that we can see here, just so that they can be pulled apart very easily. It's just for the ease of separating the DNA. So let's just take a look here. This will be our last little topic for this video, of what it looks like to go through the cell cycle. So each piece of DNA will replicate. So we have 46 pieces, and they'll all create another copy with enzymes of themselves. So from 46, double that, and that equals 92. So you end up with 92 pieces of DNA, but the 46 pieces, they all are have a DNA stuck to them. And so all 46 chromosomes will now look like sister chromatids. Okay? So number 1 through 23 from mom, 1 through 23 from dad. Every one, all 46 of those, will create a copy, and then that copy will stick to it. Okay? And so all 46, you'll have 46 of these X-looking molecules, which are actually called sister chromatids. So it's a chromosome that has duplicated, and that its duplicant is stuck to it, and that's called a sister chromatid. And these are what gets pulled apart during the process of mitosis. So we'll just run through this. You have 46 of these chromosomes, every chromosome duplicates and is stuck together as sister chromatids. And then those sister chromatids will be pulled apart in the process of mitosis. So we'll end up with 46 on one side and 46 on the other.